Hello, Pima Aztecs. Welcome to Chapter 17, Part 2, Immunization. So, in Chapter 15 and 16, we studied immunity. So, Chapter 15 was innate immunity, or nonspecific immunity, and Chapter 16 was adaptive immunity, or specific immunity. And now Chapter 17 is immunization. So, it's two sub-arms of adaptive immunity. So there's natural and artificial immunization, and then within those two categories there's passive and active. So there's natural passive immunization, natural active immunization, artificial passive immunization, and artificial active immunization. And in part 1 for chapter 17 we covered natural passive, natural active, and let's see, and artificial passive. And in this one, the whole video is going to be on artificial active immunization. So here's our table from your book. Like I said in the previous video, we covered natural active, which includes an example of being ill and then recovering from the infection. We covered natural passive immunity, including breastfeeding and IgG transfer across the placenta to the fetus. We covered artificial passive immunity, where you give a blood transfusion to this person from someone who was previously ill and already had the antibodies made. And then in this second video for chapter 17, we'll talk about artificial active immunization. That's when you receive a vaccine. So artificial active immunity, or active artificial immunity, is when you receive a vaccine. So here's a cartoon where you get an attenuated or non-lethal pathogen, or that part of the pathogen called the antigen. We've talked about that. And you put that into your vaccine, and then you inject somebody with that, and then you have to actively, so that's where the active comes in, you have to actively make your own antibodies. And then this is a cartoon, this is your blood vessel. So the blood vessel's cut out, and you can see these purple things are the antibodies. The purple Y's are the antibodies, and they're looking for these antigens to bind to. And if you want to review how antibodies work, you can look at the B cell lecture from chapter 16. So artificial active immunization is synonymous with vaccination. So it's the same thing, basically. So you give the person being vaccinated an attenuated live antigen or pathogen in the vaccine, or you give them a piece of that antigen in the vaccine. And then what vaccination does, it triggers an immune response in you, and this produces active immunity because you're actively making the antibodies, but it's artificial because you were injected with the antigen. It didn't get into you. This pathogen or virus didn't get into you naturally. And vaccination is protection from infection via a medical intervention. So vaccination is a medical intervention. So these are different ways you can make vaccines. So in this cartoon, they show a virus, and we know that this could be a DNA or RNA virus. Here's the capsid, and then here's the spikes. So you could make a live attenuated vaccine. So you could take this is kind of funny. They call it, because viruses aren't alive, right? But they call it a live attenuated vaccine. So they've just attenuated it so that it's not so lethal. So there's some mechanism to make this not a full force virus. And an example would be the bacteria Bacillus anthracis. You can give a vaccine against that organism, and the disease that you're giving a vaccine against is called anthrax. In this cartoon, again, it's a virus, and this is called a chimeric live attenuated vaccine. So basically, see this little blue piece in here? That's a chimera, because here's the viral DNA, and then this blue stuff, this is the chimeric DNA that the scientists put in to make this virus not lethal. So again, you're attenuating the virus or making it not lethal by using this chimera of scientifically made DNA and the DNA or RNA that's in the virus. And then you can take the virus here, and you can completely inactivate it. So somehow you can take this virus and inactivate it and put it in a vaccine. Inactivated bacterial vaccines include vaccines for meningitis, cholera, and tetanus. And here's the corresponding organisms associated with these diseases. You can also make a subunit vaccine, which is where you just, in this cartoon, you just take the balls of the spike off the virus and put that in a vaccine. That way, there's no virus parts in terms of the DNA or RNA that would allow the virus to replicate in your body. And then you can make a nucleic acid based vaccine. So here in this cartoon is just a DNA vaccine. So in this graph shows here 2013. In 2013, they had a weakened live type 2 polio virus in an oral vaccine, and it caused 62 polio cases. And then this board, I'm not sure what Gavi is, but it funded a polio vaccine where they didn't use a live polio virus. And then it says here in 2019, this oral polio vaccination will end, and we'll have a new polio vaccination that will not be a live polio virus. So attenuated viruses may cause disease even in healthy individuals. So attenuated viruses just means you weaken it somehow, but sometimes they can still cause diseases, so that's not the best way to make a vaccine. Here's the live attenuated vaccine that we were just talking about. Individuals vaccinated with an attenuated live vaccine will trigger the cell-mediated immune response. Hopefully you remember that's it. T-cell mediated response. And then if you get vaccinated with a live attenuated vaccine, these vaccinated individuals can infect people around them with this attenuated pathogen that provides contact immunity. So that part's kind of cool. I mean, if you can make sure that the attenuated live vaccine doesn't make anyone sick, but this virus can be transferred from person to person, this attenuated virus in the vaccine providing contact immunity, then everybody in your crew can get immunity. If you attenuate a live vaccine, or you use an attenuated live vaccine, the viruses could revert to wild type. That means to the virus that they were before they were attenuated, and that can lead to the disease. And they no longer use live polio vaccine because of this reason. And then also these attenuated microbes or modified microbes, they still could have enough virulence to make people sick if they're immunocompromised. And pregnant women shouldn't receive these kinds of viruses because the viruses could get across the placenta and harm the fetus. This is a cartoon of inactivated or killed vaccines. So whole agent inactivated vaccines, they're called that. So basically, again, this is kind of funny because this is a virus. It's not really alive, so you can't kill it. But somehow they completely inactivate it and put it in a vaccine. And then this is kind of a cool way to do it. Subunit vaccines. You just use the antigenic part of the pathogen. Neither an inactivated or a subunit vaccine can replicate. They can't revert to their old virulent cells. They can't mutate. They can't retain any of their virulence. There is absolutely no possibility of getting the disease if you use one of these types of vaccines. But because these microbes can't replicate, there's no contact immunity. And you have to get a booster shot. So here's a cartoon of stages of vaccine development for COVID-19. So everybody knows they're trying to find a vaccine for COVID-19. So in this cartoon just shows one possible way of doing it. So we've looked at this virus before, and here's the spikes on COVID-19. Remember, it's a single-stranded RNA on the inside. So and then these spikes, we've talked about this, can bind to this ACE2, angiotensin converting enzyme 2, enzyme on a human cell, and that's how the virus gets in. So what they do is they make this is called a fusion protein. So the virus relies on the protein spike to bind to ACE2. And this is the entry point into the human cell for the virus to replicate. So then they'll make a vaccine against COVID-19, and it could be in any of the ways we just talked about, or even other ways. But so they'll have their vaccine in hand, and then they make these transgenic mice that have a human ACE2 gene that will make a human ACE2 receptor on their cells so that the virus can bind to it. So then they'll give the mouse, the transgenic mouse, the vaccine, and then they'll expose it to the virus. And if that works, if that inhibits the mouse from getting COVID-19, then they'll go into phase 1 clinical trials. So they'll use a small group of volunteers for that. And then phase 2 testing, they use several hundred volunteers to test the vaccine. Phase 3, they'll do large-scale trials to evaluate how effective the vaccine is under natural disease conditions. And then phase 4, if everything works, they'll do large-scale manufacture of the vaccine, and then we'll have it. But this probably won't take, well, this probably is still 6 or 7 months away from getting a safe vaccine. Okay, so in this cartoon, it talks about whole agent inactivated or killed vaccines. So in this cartoon, we've seen this guy before, a dendritic cell. It's got its tall-like receptor. And here's the tip of the hypodermic needle with the vaccine going in. And this cartoon shows, in addition to the vaccine being injected, you inject this stuff called an adjuvant. And basically, an adjuvant, it just increases the antigenicity of this pathogen. So makes it more likely that the pathogen or the vaccine will result in making these antibody secreting plasma cells. And you have to give lots of doses of the vaccine with the adjuvant if you're doing a whole agent or killed vaccine. But because all types of these killed vaccines are recognized by the immune system as being exogenous, then they stimulate this humoral response. Toxoid vaccines. It's kind of interesting. So this guy is a toxin, and these are all the pieces of this guy that makes him a toxin. And when the scientists do chemical modification of the toxin, you can see that they made this yellow part a little less yellow. The green stayed the same, the gray stayed the same, but this big red ear was chemically modified to a flesh-colored ear. And then for the key, they say this big red ear is the toxin moiety, so that's what makes you sick. And then these guys are just antigenic determinants, aka epitopes, that will initiate an immune response. So if you're a disease like tetanus or diphtheria, you produce toxins, and it's more efficacious for scientists to design a drug to induce an immune response against the toxin, not the bacteria. So then a toxoid is just a chemically modified toxin that will stimulate the immune response, and it stimulates specifically the antibody-mediated or B-cell humoral response. You have to have multiple childhood doses as well as inoculation every 10 years for life, because they don't provide lifelong immunity. But combination vaccines like this guy, the DTaP, that you probably have heard of if you're in the clinic, is a toxoid vaccine. So this just shows a cartoon of producing a vaccine using recombinant gene technology. So this is just, again, it looks like a cartoon of a virus. We're calling it a virulent pathogen with the antigens on the outside here. And then here's the capsid with the nucleic acid on the inside. And what they do is they just delete this virulence gene, and then they put in the vaccine this harmless attenuated pathogen. It's harmless because this gene here that would make the pathogen virulent has been deleted, but you're still injecting this guy to mount an immune response. Here's another cartoon. It shows production of a vaccine. So here's your bacteria. The antigens are on the outside. Here's a piece of DNA that codes for the antigen. You put that DNA, piece of bacterial DNA that codes for the antigen, inside a yeast cell or another cell, and then that cell makes the antigen for you, and then you can put that in the vaccine and stimulate humoral or a B-cell response. Same thing, you can isolate the DNA coding for the antigen, and you can insert the DNA in, instead of a yeast or another cell, you can put it into a virus, and then now you have a virus in your vaccine that is presenting these antigens that will cause your immune system to recognize this, but this guy won't be deadly. Here's another way. So here's production of a vaccine. You have your bacteria with the spikes on the outside, and here's the DNA that codes for those spikes, those protein spikes. And then you put that DNA into a plasmid vector. Remember, those you find in bacteria. And then you make these recombinant plasmids, and then you insert the plasmid into a cell, and then the plasmid will use all the molecular machinery inside the cell to make the antigen, and there you are. You make the antigen, and you put it on the outside of this cell, and you use that cell for your vaccine. Most common problems with vaccine, there's some mild toxicity. You could have an allergic reaction to it. For example, if you're allergic to egg proteins and they made the vaccine in eggs. And then those attenuated viruses we talked about could still cause disease. And then I say here, hey, Dr. Jenny McCarthy, this child did not get vaccinated. So this looks like an old-timey casket from the early 1900s from a kid who didn't get vaccinated. There's lawsuits out there that have alleged that childhood vaccines cause autism. I think that's what Jenny McCarthy talks about. There's been a lot of research into that, and there's nothing to that, to those claims that childhood vaccine causes autism, diabetes, and asthma. CDC and the FDA have determined that problems associated with the vaccines, the mild problems associated with the vaccine, are far less serious than death. And that's the end of vaccination.